Hi, everyone. Thank you so much for popping in today. If you are joining us live, awesome. We're so excited. This conversation's going to get sciencey, nerdy, geeky, but don't worry. We are going to provide you with the tools and the resources that you need, including if you would like to get these slides and this recording all in one. I will be sending that out on Tuesday. So you just need to email me before Tuesday to Ashley@thebetternutritionprogram.com. And if that is too much of a mouthful for you, just head over into the messenger box. Hi, Archibald. I knew you'd be here for this one. This is going to be great. And if you need to, just head over into the messenger box, but do provide me your email. Otherwise, I'm going to have to track down your email from you. Okay. So what are we doing here today? So I am Ashley Koff. I'm a registered dietitian. I'm CEO of the Better Nutrition Program, and I do these Better Nutrition expert interviews. And I'll pause for a second. What is a Better Nutrition expert? Very important to define. That's somebody who understands, has experience, has worked with the science, but also somebody who is practice-based, somebody who has applied this. Because at the end of the day, science, super helpful, great information as we're going to learn about today, but also humans, very different, need to be approached individually. So we get that combination, and one of my favorite Better Nutrition experts is here with us today for a topic that really needs a Better Nutrition expert. So Amanda Archibald from the Genomic Kitchen is here with me today. And if you are watching this on a replay, if you're watching it at a better time, or you start watching us and then you have to go somewhere, or the slides maybe don't work in a moment and we're figuring all that out, just kidding, that wouldn't happen. But if any of that goes down, I want you to know that if you have any questions, go ahead and drop them in the question box here on Facebook, or if you're watching on YouTube or you're watching at that point, just hit reply to whatever email you received it from. But also understand that we are just going to scratch the surface here today of Amanda's resources. She has an amazing book that I spent my summer reading through. It didn't take me very long, but I went back and I love it. It's easy reading, but it helps really understand culinary genomics. And it comes out 2 days after my birthday on September 10th. So you can head to the Genomic Kitchen, and in the book section, you can tag that. Araceli says, hi, Amanda. And so you'll be able to get that, but mostly in complement to today as well, you can also get her free guide. And if you're a practitioner, you can check out the courses, which is on my to-do list because I'm really excited. This is one area that I know I need to learn more about, not just to practice, but to express recommendations, whether on social media, or whether in the media, or whether even just how to understand what science may or may not be telling us these days. So a lot of different applications. So that's my mouthful. All right, Amanda, we are here, and what we were just giggling a little bit about is that we love science, we practice all these things that we are great at. Skype, eCam, and pulling up slides, we'll see. So this might be a little bit clunky, but I've done it before. Once we get started with Amanda, we're going to pull up the slides, and we're going to go through that part. But if you have any questions as we go along, go ahead and interrupt me. I should be able to see your questions, and we'll get there. And if not, we'll get to the questions at the end. Okay, Amanda Archibald. Why MTHFR? What are we talking about today?
So I think someone summarized it in one of my professional Facebook groups by basically asking the question, MTHFR, is this the new gluten-free? Because everyone seems to have it, right? And I thought that was such a great statement. And it is true that everyone has the MTHFR gene. The question is, what does it mean? Mm-hmm. Right? And so today we're going to talk about what is MTHFR, what is its relevance to human biochemistry and our health, what do we need to know, but what else do we need to think about? So that's where we're heading, because we want to recognize its importance, but we also want to downplay a little bit of the hysteria around it, right? So that's where we're going, because you and I just flip back and forth on this, and I said, let's get this straight.
Mm-hmm. Right. So today we're talking to practitioners. So those of you that are practitioners, hi, we're excited to have this conversation. And we're also talking to individuals, hi to all of us who are individuals and patients ourselves, to give guidance on that part. So again, if the science goes over your head, don't worry about it. We've got plenty of resources to help bring it into your head and into action. And we will talk about those in conclusion. But what I'm going to do right now is I'm going to flip us over to the slide space, and I'm going to get started, and I'm going to cruise direct Amanda through this. this just in case she's not able to see it as easily as we'd like. All right, here we go.
Right, so this will be fun because Ashley may be able to see the slides, but I may not. And so I'm going to pull them up. Did you pull them up already?
I did, and I'm on the home page. How you doing? Can you see them or you can't see them?
I can't see them, but I'm just going to do them right off of my screen. So if any of you were looking at me and you're like, why isn't she looking at the camera? It's because I have to look at my screen at the same time, say, Ashley, this is where we are. Change the slide, change the slide.
Yes.
But both of us usually work on Zoom, and so you have full control. So anyway, we'll just kind of roll along. And so here we have our starting slide, understanding MTHFR. So what does it mean? And the reason we abbreviate it is because here's what MTHFR stands for, methylene tetrahydrofolate reductase. How about that? It's a good job that we're not doing a biochemistry exam, right? Because even being able to spell this, right? But what we want to do today is get deep into MTHFR and put it in context. So I'm going to move on to my next slide.
We're there with you. Yep.
You're with me. Okay,
yep.
So what are we talking about? So MTHFR is a gene. So we're just going to go right back to, well, what does a gene do? And so, because I always think it's important to put everything in context. So the thing that comes up so much about MTHFR is, first of all, what is it? What does it do? And how does it get kind of affected? So how it gets affected is in the way that the gene is produced. So we're going to go into genomics. Some of you have seen this already if you followed my work. Some of you know yourself. Some of you maybe have no idea what you're looking at. So that's what we're going to look at. So basically, a gene is a section of DNA. So that's what we're looking at the top. So DNA, as we all know, is the instructions that we receive from our parents that build us as a human, right? And as humans, we all share pretty much the same genes, like 99% identical. Otherwise, we'd be mice or rats or donkeys or something other than a human. But what comes up is, well, how do we get to be different? So because we have both practitioners, clinicians, and members of the public with me, I'm going to kind of step this back and make it pretty simple, just so we're all on the same page. So the bottom line here is that genes are responsible for making proteins. So the way that we make proteins is we actually use a specific code, which we'll get into in a moment, because I'm going to show you binary code for computer code versus DNA code. So at the top, what you're seeing is the DNA information. So you may want to just point that out, Ashley, where it says DNA. And those, what you see there is you see ATG, ATC, TCG. Basically, our DNA is made up of 4 base, what we call nucleotides: adenine, guanine, thymine, and sister, whatever. I forgot what it is. It's like what I'm broadcasting, right?
That's right.
So ATC and G. It's funny when you say something every day, then suddenly you're like, what was that?
Yeah.
Anyway, ACCG, that's what we need to know. So basically, DNA, our genes basically will hand off the recipe for proteins to that intermediary where you see RNA, and that process is called transcription. So the gene's job is to hand over the recipe for proteins, okay? Proteins, let's define them, as we all know, are the construction workers in the body, and they provide the infrastructure for the body. So we like to think of it as a building. Like proteins are like the scaffolding or the infrastructure, but they're also the doers, like the construction workers. So they have a dual role. So your genes, each gene has a very specific recipe for a protein, and its job is to hand it off to messenger RNA. That's called transcription. And then when you go down to the bottom of the screen, you see on the left-hand side, translation is when that RNA basically converts that recipe into a protein. So that's what you're seeing at the bottom. So those of us who went and studied biochemistry, you're recognizing those as the fundamentals of protein, which are amino acids. So methionine, isoleucine, and serine, we basically cinch together all these building blocks of protein to make a protein or a polypeptide, right? So basically you're seeing the information processed through to provide a protein. For those of you who are not involved in biochemistry, think of a protein like a pearl necklace, where each pearl is an amino acid that's strung together when we make a necklace. The question is, does your pearl necklace look like mine? And probably not, because no one pearl is identical, right? So everyone's going to have a slightly different pearl necklace. And that slight difference in how we make up that pearl necklace is actually in our genes, is actually a slight error. And it's those slight errors that make the difference, not only in our traits, what we look like, but it also makes the difference whether a protein like MTHFR actually functions efficiently or not. So that is an important part to think about. And if you go to the next slide, Ashley, let's put that in context. So you see the slide says, another way of looking what Amanda just said is, wait a minute, humans all have like 99.9% identical genes, but we don't all look the same. How so? So let's put it in context. And I love this slide. This was this illustration. It was created by a medical illustrator. She's also a dietitian for me. So what you're looking at the top is what is binary code. So I was lucky enough in school. Actually, my math teacher actually taught us binary code way back when, and we had no idea what we were learning. But he was smart enough to say, you're going to need to know this for the future. So the bottom line is binary code is made up of zeros and ones, right? And we string those zeros and ones together in a string of 8 digits. And then we put those bits are then put together in a byte, and we make a word. So each of those lines corresponds with one letter in the alphabet. So we create a word, right? So in our word at the top, it says engaged. So if I were to say to you, hey, my 2 best friends got engaged, how cool is that? They're super happy. You'd say, oh, that's nice. What if I had a slight slip of the tongue? I said, my 2 friends just got engaged. They're really happy. Isn't that nice? You say, wait a minute. Did she mean engaged? She said engaged, but I think she means engaged. Yes, she means engaged. And you don't want to, you go on and you don't think about it. The same thing can happen in the human body, that you can have a slight like transcription slip of the tongue, and the body says, that's not quite right, but we get what the protein's trying to do, makes no difference. Or you can have a slip of the tongue, you're like, that doesn't quite right. So let's look at it in DNA code. So if you look at the bottom, you can kind of circle that, Ashley.
Yeah.
What we're looking at is the impact of one of those errors, slip of the tongue, in a gene. And so if you remember, the hereditary material is A, C, G, and T, and there's all kinds of different formulations. So in this case, we're making a protein, and the second amino acid in our protein is threonine, right? Threonine, so that's an amino acid. And what we see on the right-hand side is, hey, this is what this protein looks like with threonine in this position. If you go down to the lower, the lowest picture, what you see, again, we're just really illustrating the point that will bring us to MTHFR because it's important. What you see is we no longer have ACA. We have CCA. And so we have a different amino acid now in our chain, or in our pearl necklace. We've got an aberrant pearl, right? And when you look at the protein, it's a different shape. So what does that mean to the body? Depending on the degree of change in that shape can affect how that protein functions. And proteins, what they have to work with cofactors, which we all know are vitamins and minerals for the most part, right? And when we change the shape of the protein, it changes how those cofactors bind, which means they may not bind to help the protein do its work. So it's like putting brown sugar instead of white sugar in your cake mix, just to be, really, really simple. In some cases, it makes a difference. In other cases, it tastes the same, looks the same. Cake may be a little browner on top, slightly different in texture, but it's still a cake. It's the same when we produce any protein. Sometimes it can have an effect. Sometimes it's minimal. And that's what we're looking at when we're looking at MTHFR. What's the degree of the impact of that gene or the protein, the enzyme it produces? Does it have an effect or doesn't it? And what happens is people say, oh my gosh, I've just learned through my testing, my DNA testing, that I am homozygous MTHFR, which means, and we'll look at that in a moment, it means, oh gosh, that means that the shape of this protein has changed completely. That means it's not going to work properly, which means I'm going to have all these effects. I'm doomed. And what we have to look at is the entire picture of not just that gene, but how it functions with other aspects of our biochemistry. And this is what the most important part is. And Denise Furness, our colleague that I just introduced Ashley to, she's a PhD molecular geneticist with a master's degree in nutrition, too. Brilliant teacher. I've had the opportunity to work with in Australia. She said it very clearly recently. You cannot be diagnosed with MTHFR.
Right.
But you can be diagnosed with some of the impacts or insufficiencies that result from having a slight change in the enzyme from MTHFR.
I love that, Amanda. I love that one because I have a lot of people that are like, "Oh, I have MTHFR," and you're like, "Oh, I have eyes." Like, get it? Like, gotcha. Like, it's like we now need to understand. Maybe it's the color of your eyes, or it's how you see, or we're looking at all of that part. But I think that's such an important one. Absolutely,
it really is because really the impact of MTHFR is whether it impacts how much folate you have available, which we can make a workaround, and your homocysteine levels. And we're going to look at that and put that in context. So you can't be diagnosed with MTHFR, but you can suffer some of the consequences of it not working efficiently. But guess what? The human body is brilliant. And when you work with clinicians or practitioners who understand how this gene works and what we need to test for, we can create workarounds. It is not a sentence for anything, but it gives us a clue in the toolbox is what we need to look for and what we can correct through food. So,
Amanda, I have a question because I remember back in my training days, I was working with individuals who had metabolic diseases where not a mutation or an alteration or, looking at the profile, but where they were actually missing that gene. Is that something that is diagnosed? Like, could it actually be accurate that in a small portion of a population that there would be somebody who literally doesn't have the MTHFR? Or are we pretty comfortable as, saying across the board that most individuals have MTHFR and it's just really a question of what their MTHFR is doing?
Yeah, it's a very good question. And what you bring up, first of all, I don't know the answer to whether there is anyone that's born without the gene. I've never been asked that question and I don't know.
Yeah.
But I think what you're bringing up is we have to distinguish between what you see. So change slide to the next screen so you see three people on it.
Yeah.
We have to distinguish between what we call single nucleotide polymorphisms or SNPs. So you can see that on the left-hand side. And mutations. So SNPs, we all have them. A SNP is that little spelling error. So if you look at the three people on this slide, you can see they pretty much all have the same code, right? But what we're showing in the colored area where it says SNP ACE-GT, what you're seeing here is the difference between these people is a substitution for one of those base nucleotides, which means it's like putting the proline amino acid instead of the threonine. This is what creates the difference in our biochemistry. This is what it explains some of this, why, you can eat all you want. And you let me give you a better example. You and your sister or your sibling eat exactly the same food, exercise the same, but you have different metabolic outcomes. One of you has to work harder to build muscle or maintain weight, gain weight, lose weight, whatever. This is what explains that. But one SNP does not kill you, at all. That is very different from a mutation, which is a little bit the area you were looking at.
Right.
But it's not exactly the same thing. So a mutation appears in less than 1% of the population, and it usually is fatal. It will, over time, create some kind of a fatal disease. There's a fatal outcome.
Okay.
And this can be managed by medicine. However, it can severely restrict lifespan. So you're looking at things like phenylketonuria, cystic fibrosis, etc.
Okay.
And the difference between SNPs, and this is very important to MTHFR and a genetic mutation, is that a SNP or a series of SNP will respond to food / supplementation, lifestyle modifications such as exercise, stress management, removal of toxins. Okay, that's point number one. So mutation is very different. occurs in less than 1%. SNPs, we all have them. The SNPs become important, as we'll see a little bit later on, is it's not just one SNP. This is important for MTHFR. It's not just whether MTHFR is functional or not, its enzyme. It's all the enzymes around it. So what we do with this kind of genomic information is we look at the pathways and processes that a SNP impacts. That's what's important. So when somebody says, so even with us as clinicians, you know, some of our clinicians are new to this area. It's like, oh my gosh, somebody walked in and said, I'm homozygous MTHFR. And the first question I have is, what laboratory information do you have? Or what other genomic or genetic information do you have? Because you can't just look at one gene, right? It's wrong. You know, if we're just ordering for MTHFR, it's like, okay.
Yeah.
It's a much bigger picture. So hopefully that answered your question.
Yes.
It's a good one. It's important. All right. So we all have single nucleotide polymorphism. So let's look at MTHFR. Okay. I've got to kind of move you off my screen here. There you go. So I can look at my slide. So here we have a slide with on the left, it says MTHFR 677 and MTHFR 1298. These are variants of the gene. So when we just looked at those SNPs, a gene comes, as Ben Lynch says, it comes in different flavors. So a gene has some different faces to it. There are many, many different variables of MTHFR, but these two, as you and I know, Ashley, are the ones that are most studied. And when we're looking at evidence-based science, and as we translate this, it's really important to look at the degree of evidence. So I'm hearing now, I actually heard the other day from a genomic testing company, they are actually testing even more variants of MTHFR, and I don't yet know what that science is, what the level of evidence is that I would use yet. But I'm learning like everyone else. So we're going to talk about MTHFR in context here in a moment. So we're leading up to this. So MTHFR, when somebody says, I have MTHFR, well, yeah, we all do have. What's important about it, and the reason, just flip back one slide for me, Ashley.
Yeah.
What's important, I have to flip back my own slides here. What's important is, and you can probably highlight it, see where it says SNP and we have AGT. What we're interested in is where these substitutions are in the protein recipe. It's those substitutions that tell us how much that is impacting the enzyme. So remember that, and I'll go to the next slide here. So what we know with MTHFR is there's two, as we said, the 677, the 1298. These are the most studied variants of the genes. And where it says enzyme activity at the underneath, it's the combination of those substitutions, whether GCAT, it's which substitutions that appear with these two genes that will dictate the degree of activity or loss of activity of that enzyme. Does that make sense to you, Ashley, as I presented that?
100%. And I think it's so valuable to say it exactly that way. So I'm just going to ask you to repeat it one more time, because I think practitioners need to hear it articulated that way. Yeah.
And you as the public too. Because, and this is what genomic testing companies do. You know, it depends on the company as to how they output their information. The companies I like to work with show me this, and they show me the level of evidence. So with the enzyme activity, where you meet, and this is rare. It was like, I don't even know if we see someone who has no enzyme activity but with the MTHFR 677, it's the CC variant, and the 1298, it's the AA variant, very rare, that would say this gene doesn't work at all, so you know, the protein is useless. Very rare, right? The CCCC, you may see a 60 to 70% reduction. The CT and AC is one we're probably more familiar with. Could be up to a 50% reduction in the enzyme's ability to function. This is what's really important. It's the degree to which it is or isn't functioning that's what's important. But the great thing is, regardless of whether it's functioning or not, we can still create a workaround. And that's, I think, where we're going to get to next, Ashley, is like, okay, let's put this in context, because we need to get this right once and for all. So hopefully everyone can see that. And like, even if you're fluent in this language, those of us who are, we go back and say, wait, what?
Yeah.
What was that? But the bottom line is we're looking at functionality. So let's jump to the next slide, as I will.
Yep.
Okay. I'm just picking up my chair rest here.
Yeah. Leafy greens. Here we go. Leafy greens.
People, do not freak out. Some of you are fluent. Others are like, oh my God, what am I looking at? But I think as I go through this, it's going to make a lot of sense because it's super, super important that we understand this. A big shout out, this slide, and a couple of them today come from
hang on. Amanda, I'm just going to interrupt. I just want to, Shana, I apologize if I didn't pronounce it correctly. She just wants to confirm. So if we see 50% on the last slide, she's saying that the question mark, and I want to make sure that there's clarity, is does 50% mean it's functioning, the enzyme is functioning only at 50%?
Yeah, it could be it's a degree of efficiency.
Uh-huh.
But understand, and this is what I'm going to show you, that even if there is a loss of efficiency, there's ways we can work around that. What does the loss of efficiency mean? Okay, and that's what's important. So it's a really good question. MTHFR, even if you've lost 50% of efficiency, is not a death warrant at all. It's a signal to those of us who are clinicians to say, okay, let's look at their folate. Mm-hmm. So I'm going to show you that. This is, and does this person get a lot of folate from the diet? Super important. And if they don't, how can I shore up or create a workaround, like a diversion, or like how can I go on the frontage road because that gene isn't working efficiently? This is where it gets to be interesting. Okay, so hang in there with me. So,
we got it. She said thank you.
Actually, you have a lot of experience in the supplement area here, so you can definitely weigh in here.
Yeah.
So what are we looking at? Basically, MTHFR, and we're going to kind of go in a number of different directions with it, but let's look at its functionality. MTHFR is really, really important in that it basically generates a product called 5-methyl tetrahydrofolate. Mm-hmm. Okay, so that's what it does. But guess what? So you can see this, that folate comes in a number of different forms. So it comes in a number of different forms from the diet, which have to be converted. And what's important is to look upstream. So if we look upstream here, there's a number of different enzymes, and these are all encoded by genes. So what your question you ask yourself is, if this gene is working 100%, but these genes are not, then you've got a product that's not even presented in its right form to MTHFR to do its job.
Right.
So when somebody says MTHFR, oh my God, I'm homozygous, I'm like, okay, what else is going on?
Mm-hmm.
So folate, let's go back. Folate, which we get think of foliage, right? We get in a number of different forms in the diet. Okay, and the form that we get from the diet that we need that's presented to the body is in the 5-methyl tetrahydrofolate form, because when we take in folate in leaves, let's say we're eating our kale, the bacteria in your gut can actually convert it to 5-methyl tetrahydrofolate. So MTHFR is not relevant. All right, so the number one thing, now, okay, if you have dysbiosis, that's a different issue, right? Okay, if your gut is not in working order, maybe we're not able to synthesize 5-methyl tetrahydrofolate or convert it to the form the body needs it from the gut. The human body is brilliant. It's biochemically brilliant. Sometimes I look every day and say, how do we even function? It's that genius. So the number one thing is eat food, whole food, uncontaminated food, lots of vegetables, and this becomes sort of less relevant, right? Does everyone see that?
And I think, Amanda, I'm gonna interrupt for one sec because I think there are like 3 things that pop to mind when I'm looking at this, and I want to just make sure that and it so underscores what you're saying. Like, we know there, and it actually is just easier on the body if you take in leafy greens, right? And we know the rest of the country is starting to learn that, and we're moving in that direction in a lot of ways. But we also know that our country took an initiative in the 1920s to fortify everything with folic acid. And so, a lot of like I have a patient who was saying like, oh, my folate intake is good because, and she actually was doing, I can't remember, it wasn't a chronometer. I think it was a MyFitnessPal or something, but it was like she was getting credit for that because she was having fortified, breads, crackers, like those kinds of things. So I think one thing we have to talk about is, as you were saying, like if you just eat whole foods, but we have to remember too that that fortification. So that's one piece.
We're going right there.
Yeah. And I think this second piece that I think is just so valuable when we talk about dietary supplements. So if we take the food conversation that we're going to have, and then we're going to have the dietary supplement conversation, or just to be thinking about it through this lens, what we are looking at right here is such great evidence about why supplements are often not effective and even sometimes harmful in your patients. So, you know, you'll see that like SAMe is recommended, or betane's recommended for something, or folic acid, or B2, or B6, or any of these things. But when you see those recommendations, you've got to know both the format and also, is the rest of the diet, like, is there total nutrition supporting all this? So that's just, when I look at this slide, I'm like, wow, this tells me everything I need to know. Yeah.
Right.
Yeah.
So, on that point about folic acid, let's get there.
Yeah.
So you and I were talking about this the other day. I think if there's number one major recommendation I would make right now for anybody, whether you're listening as an individual interested in your health or your clinician, is to go down to your pantries or look at your supplements, and anything that has folic acid in it, get rid of it. Here's why, and this is a huge learning curve. Some of you may be fluent in this, is that folic acid, I think, Ashley, correct me if I'm wrong, but it was placed in flour. Basically, anything that was made from flour was fortified with folic acid because we were trying to deal with anemia. And it turned out that I believe that folic acid in its form they saw a response to anemia. But this is, like you said, in the 20s, 30s, maybe the 40s in Australia, that we didn't know about methylation. So we're going to come to methylation. So folic acid, it kind of worked, right?
Well, it was actually, and yes, and I think the macrocytic anemia, but the primary driver was we couldn't figure out what was going on with neural tube defects. Okay, and that has a ton of relevance to this. And you're right, absolutely, on the anemia part. But because one of the places that this is so extra special, when you say go look at your supplements, but I would even argue go look at your protein powders, go look at your crackers, your kids' cereals, you know, all of those. The bars, like a lot of the packaged bars that just drop a multivitamin into them. Yeah. Take a look at those.
Folic acid's in there. And this is and let's really get into this and then move this through because it's super important. Folic acid is a synthetic form of folate. And if you would put the two molecules next to each other, they look absolutely identical, except folate, or this form of folate contains this methyl group. And we'll talk about what's a methyl group in just a moment, just so we're all on the same sheet of paper, which means the folic acid is synthetic. It's inert. It's not biologically active from the diet. The body has to convert it. And the way it converts it is through this dihydrofolate. Okay, so it turns out, and this is what you learn when you dig. So it turns out that when they were looking at creating folic acid as a supplement, of course they tested it in mice, and mice have a very, very efficient DHF enzyme. So it could very quickly get folic acid into the cell. Great, we've achieved it. In humans, this works very, very slowly, which means that if you have folic acid in the supplement form, we're talking about folic acid, not folate. If you have that anywhere, it's in your diet, supplements, whatever, it will snarl up this enzyme. So in other words, it's kind of hanging out there. And you say, okay, well, what does that, so what? It absolutely saturates its enzyme. The problem with that is then you have elevated levels of folic acid floating around the body. And to get folate into the body, into the cells to do its job, we haven't even got to what its job is yet, it has to bind to receptors. So you've got folic acid. There's a lot more members on the folic acid team than there are on our 5-methyl tetrahydrofolate that we've got from food if we're fortunate, right? Which means 5-methyl tetrahydrofolate cannot bind to folate receptors to get into the cell to do its job.
Right. And I think like that to me like is so standout because I've been in great arguments with healthcare practitioners and with scientists, and also to be very fair with people who are concerned about and I think we all are, but about access to resources in the country, where we're saying, okay, but like folic acid isn't harmful. It's still a better way, and especially if somebody can't afford methylfolate as a supplement. And what we're saying here is no. What we're saying here is, you don't have to eat organic greens, like just eat leafy greens. But don't get in the folic acid, especially based on, a lot of when you play that theme out of what do you have access to, if somebody is if the rest of their diet is nutrient poor, the likelihood as well of the gut health and all of this stuff. So I just think what you're highlighting now is really game changer in the way that recommendations are made across the population.
It's critical. It's an socioeconomic issue. It is a huge issue because why is MTHFR, why is it such a standout? Because it's been associated with cognitive, mood disorders, mental health. There's fertility issues that are emerging, et cetera, et cetera, et cetera. So, obviously we can spend a long time talking about that, but let's talk about 5-methyltetrahydrofolate, what it does, and really get into this mechanism. So if we could actually convert folic acid, it would have to go through a number of steps. Then it's presented to MTHFR. MTHFR will then convert that form in conjunction with riboflavin to 5-methyltetrahydrofolate. If there are SNPs or disruptions to any of these many genes, and I'm not even showing you all the genes here. That's on the next slide. Just to see you. There's many, 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 many steps before we even get to MTHFR. So we want to know, I want to know clinically with a client, how are they utilizing folate? Can they utilize folate if they don't have any supplementation? And if I see a number of errors in these genes, I'm like, you know what? With this individual, if we can't push 5-methyltetrahydrolate through the diet, then yes, I would recommend that they supplement. Why do they need to supplement? Why do we need 5-methyltetrahydrofolate? So many different reasons. What you're seeing over here is what we call the methylation cycle. So it's methylation madness. We want to understand what that cycle does, right? Which we will. What that cycle does is it creates something called methyl group. So a methyl group, just so we're all on the same sheet of paper, is a carbon surrounded by 3 hydrogens. The methyl group activates different molecules around the body over 200 different reactions. So it's like it has the big key to so many metabolic reactions in the body. We'll actually look at one in neurotransmitters, so in brain health here very, very shortly. So we want this cycle to work. What 5-methyltetrahydrofolate does is it gives off, see the methyl, it gives the methyl groups off to this compound, homocysteine, because homocystine's job is to recycle this important amino acid, methionine. Methionine then gives its methyl groups to this molecule, SAMe, S-adenosyl methionine, which is the master of the universe of handing out methyl groups everywhere in the body. Okay, so this is a very important compound because it's how we create methyl groups. So the folate cycle is really, really important. But guess what? There's rate-limiting steps. And the rate-limiting step, which means there's a catch, is there's a couple of genes here. We're just showing one. It's called methionine synthase. Methionine synthase requires B12, methylcobalamin, to do its job. So this is where I say, okay, you've got 5-methyltetrahydrofolate, but if you don't have sufficient levels of methylcobalamin available, guess what happens? You get stuck here. This is actually what we call the methyl trap. It means folate just gets stuck here, which means this cycle gets backed up, which means you don't create these important methyl groups that need to do the job. We're going to look at how they're going to do the job in a moment. So just hopefully everyone can see that.
Yeah, and I've been doing a little dance with the arrows. One of the things I think, and you mentioned it before when you said riboflavin, just because riboflavin isn't noted on here, I think we were often taught, I think maybe it was one question or one course part where it was like homocysteine, B6, B12, folate. But I just think, do you want to pause for a second and talk about riboflavin, B2, just its role real quickly so people can see where that is mapped out on here also?
Basically, for this gene, this MTHFR, the enzyme needs B2 as a cofactor to work. So sometimes you'll hear people say, well, just shove in some B2, some riboflavin. What happens if you add in a bunch of riboflavins or a B complex, but we haven't adjusted for the B12? Where do we get B12 from? From the diet. Can the gut synthesize it? The idea is maybe it can synthesize this active form of B12, but even if it could, the levels we need would exceed the ability of the gut to produce it. So for this cycle to work, you need, this is where the B vitamins come in, the B6, the B2, and B12 to work. So that's when people say, I have MTHFR. I'm like, okay. So let's look at, we're looking upstream and we're looking downstream. So a lot of times when we're looking at whether these cycles work, we may measure folate, we may measure B12, but we may also be looking at these intermediaries. And this is where I would always say, as our colleague Denise Furness said, you can't be diagnosed with MTHFR, but you can be diagnosed, or you can be told that you have low levels of methylfolate or folate, you have elevated of homocysteine, and sometimes B12 often could be high, and there's a lot of controversy around the sensitivity and specificity of measuring B12. So a lot of times people say, well, I have a B12, high B12, and I don't know why. And it's like, actually, it's because you're deficient. It's not because you're efficient. So we look at folate, we look at homocysteine, we look at B12. Sometimes we can measure these other intermediaries. That's the picture that we look at. MTHFR will just tell us, okay, let's see what isn't working. And when you have all these biomarkers or these laboratory markers, then you know what to do through food, right? Say, okay, let's get the right information. Let's correct this. The question is, why do you need to correct it, right? Because all we talked about is methyl groups. Why do we need to correct the methylation cycle? Because those methyl groups do impact a number of different areas. And one of the things you can see here, this is a methylation cycle. Homocysteine, we don't want it to have elevated levels because elevated levels of homocysteine are associated with heart disease, right? And it's because it acts as a pro-oxidant. So basically it can damage our blood vessels, the endothelial layer. So it acts as a pro-oxidant, so creating a lot of oxidative stress.
So Amanda, a question for you, for those who are watching who this is something they want to apply or this is something that they want to apply in their practice or apply personally. Let's say that they watch this and are like, okay, first thing, I'm going to go remove all that folic acid. Like I'm going to go look at my cookies, my crackers, my bars, the stuff and my supplements. I'm also going to make a real effort to eat the leafy greens. I got tested and my B12 and my folic acid, things look good there. I don't have elevated homocysteine, and I'm switching over to methylated formats of methylcobalamin and methylfolate. And we'll talk about all of this. But what's the time frame in a healthy person that you would expect to see that, I shouldn't say that. Let's not use a healthy person for right now. Let's say that we're working on somebody's homocysteine. What's a time frame of making these swaps over that is realistic for a practitioner to say, hey, or to for an individual, go back and get your homocysteine levels checked?
Yeah, we're looking, it's a very good question because we have to look at bio-individuality, right? Right. If you're correcting dysbiosis at the same time,
yes,
that's completely different than somebody who you're just kind of nipping and tucking, who, is maybe looking at getting pregnant, and they really kind of prep themselves. So I like to think of about 3 months.
3 months. Okay, great.
But be realistic that what you're trying to correct, because if you know that B12 can be the active one can be synthesized somewhat in the gut, that's out of the picture until you reestablish the efficacy of the gut. So,
and that makes a ton of sense. That's kind of like cholesterol. So I think just a good, a better, not perfect operational marker for people would be like you would retest your cholesterol levels at 3 months, you could consider retesting the homocysteine. And of course, when it comes to cholesterol, when it comes to homocysteine, to any of these, you would have to have tuned up your digestion. You would have to almost add those on as extra time before you think of those 3 months. But that's super helpful. Thank you. Okay.
No, you're welcome. One of the things that's really interesting here, and we're not going to get too deep here, but I just want everyone to, we're talking about MTHFR, right?
Right.
Hopefully everybody's starting to see, That's just one element in a much bigger cycle. If you are vegan, there are plenty of healthy vegans, but methionine is an essential amino acid, right? Can you see how important it is? This is the jump-off point so we can create these methyl groups. Methionine is often deficient in vegan diets because it's predominantly found in animal proteins. It doesn't mean to say you can't find it in other non-animal products. So if you look at sea vegetables, particularly spirulina or Brazil nuts, are an excellent source of methionine. Excellent in terms of how they compare to other plants, but diminutive in terms of how they compare to animal products. So it's something to pay a lot of attention to. Methionine, and let's just talk a little bit more. I want to jump in here because a lot of people are very interested in keto, right? So keto, the ketogenic diet is a higher fat content. It depends how you're manipulating it. So to properly metabolize fat and to basically extract the energy from it, we need a molecule called carnitine. So carnitine helps move the fatty acid residues across the mitochondrial membrane to the electron transport chain so we can produce ATP, right? So we can produce energy. Guess how carnitine is produced? Methionine and lysine. And methionine gives 3 methyl groups to lysine to form, I'll make it very simple, carnitine. Can you start to see here that if this doesn't work, this is where if you don't have the methylated form of folate, or if B12 is deficient, or maybe we don't have enough methionine, how that could affect how you metabolize fat. Because you need carnitine as one with our long chain fatty acids, with our saturated fat. It's all connected. So that's one example right there of where you sometimes may see people who have elevated triglycerides or non-alcoholic fatty liver disease. It's, elevated triglycerides, they don't know where to go. Lipid abnormalities. It may be as simple as looking at these 2 cycles and making sure you have these base nutrients to form the molecules that we need to get the job of our biochemistry done. I think that's an amazing example. So when somebody presents that to me, and I'm actually working with somebody in Australia right now who said, look, I have non-alcoholic fatty liver disease, which we're seeing significantly in the United States. We're like, why? I am postulating already that she'll have some aberrations in her folate but B12 metabolism exactly for that reason. We'll see. And one of the things here I just want to point out, as soon as we have clinicians here, is this betaine the betaine is also a methyl donor, and so is choline. So choline in eggs, betaine you find, this is where botany comes in your Chenopodiaceae family, for those of you who are gardeners. So beets, amaranth, quinoa, all in the same family, are an excellent source of betaine. So sometimes when we have a little bit of a snarl here, there's a frontage road. The frontage road is using products that are rich in choline or betaine that can actually act as methyl donors to make this reaction work. Biochemistry is beautiful, isn't it? All right. So am I making sense, Ashley? This slide is really important.
I love this slide. I feel like I honestly, I know there's so much that we're going to go over, but I honestly feel like this slide and what you've shown here is also just the strongest argument for it's not take a bunch of methylfolate and call me in the morning. Like we've got to do it differently. Yeah.
Our biochemistry is interlinked. I like to think of when we used to go to like Disneyland or whatever, you get on those rides where they zoom together. Like, I don't know what they are, but this is your biochemistry. It works like, these rides, the cycles that collide with each other. The last thing I just want to show on this slide, and let's get off this, I also want to acknowledge this slide comes from fxmedicine.au, so FX Medicine Australia, great website for the public and for clinicians. So this is actually what they call monographs, so just they're very good illustrators. The only other thing I want to show is actually under leafy greens where it says folinic acid. So that's a form of folate that is also in a supplement form. And it's often given, and you would know better than me, it's often given it has another name when we're treating cancer. Why is that? Because this form, the 510 methylene tetrahydrofolate, is actually instrumental to the production of thymine, which is one of those base nucleotides in DNA. So when you hear that folate is involved in DNA synthesis and repair, 5,10-methyl methylene tetrahydrofolate takes uracil and converts it into thymine. So thymine is then available for DNA synthesis. When we know that folate is deficient, we can often look at the uracil to thymine ratios. And if uracil is elevated, it's one of the markers of DNA damage versus DNA synthesis, because it requires this folate molecule to make that jump to thymine. So that's why that's often given. It's often given, I believe, it's not my area of expertise, it's a specific form when you work in oncology, because we try to repair DNA, right? And we also need to grow cells, but not to replicate the wrong ones.
Right.
It's also a form of a supplement. You could see here in this cycle, you're jumping through some of the slow genes, or you're jumping through a lot of genes to get to right before MTHFR, say here. Here's just convert 5,10-methylene tetrahydrofolate to the 5-methyl form, and off we go. So on that point, let's go to the next slide, which we're going to spend like 1 minute on. All I'm showing you here is just how many steps there are before you get to MTHFR. So when clinicians say, oh my gosh, somebody came in their home, I got Zygous MTHFR, I'm like, okay, what's going on with these other genes? There are so many other genes that we consider to look at the picture. You know, and that's what's really, really important. So we're not going to get into the deep parts of all of this, but just to say MTHFR is just one part of the picture. The other thing I just want to show here, and we'll get a tiny bit more into it, is on the left-hand side, you might want to cycle out. See there's 2 MTHFRs, there's another MTHFR down there. It's a gene, just cycle that. What you're seeing to the left of there is MTHFR is very, very clever in that it directs traffic in a number of different ways. The first one, of course, is to create the methylated folate, which is handed over to homocysteine in the methylation cycle. That's on the right. But the other thing it does on the left, super important, and this is like the brain connection, is it actually helps recycle the molecule. You can see BH4 at the top of that. From BH2. BH4 it's part of the biopterin cycle, That is the cycle where we produce neurotransmitters. The food, the mood, the dopamine, the serotonin, and also norepinephrine and epinephrine, so the catecholamines. So MTHFR plays a role in helping that cycle work in a couple of different ways. This is one way. It's directly recycling the BH4 molecule. And the other way we'll get to in a couple of slides. So just move on to your next slide.
Can I ask a question on that side real quickly? Where we've got the folic acid up at the top, is that more of a discussionary point of like saying like, hey, here's how folate would track through? Or is it basically saying,
these are the number of steps that if you were eating folic acid,
right,
it would have to go to, and see that that dihydrofolate molecule. That is the one that's slow in humans. Right. So folic acid technically could actually go through all these steps, right? So that's an important point.
Right.
It could go through all these steps, but it is so slow. And if you did have SNPs in any of these genes, could you see how you wouldn't get to the methylfolate from folic acid?
Right.
Plus, as you know, it can be toxic because it can end up just hanging out there. And I think I heard a couple of researchers the other day say every single one of us has unmetabolized folic acid floating around in us.
Right.
Because if it's just ingrained in our culture through the type of food we're eating, through every restaurant, what's the first thing they put on your table? You want bread? Right?
Pun intended when you say ingrained. Because it's in the grain. There you go. All right, chuckle.
So folic acid technically could be metabolized, but it can snarl, and the levels that are being recommended, what is it, 400 milligrams? Folic acid can actually be I'd say toxic, can overwhelm the dihydrofolate enzyme at about 200 micrograms.
Right. And especially in prenatal, too, when you're saying 600 or 800, and what if you don't have healthy digestion, and what if you have a SNP? So, yeah, miserable. Okay, thank you for answering that one. I'll push us onward.
So, yeah, so next slide. So we've already talked about this next slide, but those of you can kind of mull on it. Again, this is an illustration from fxmedicine.au. I just want to, because I always want to acknowledge where information is coming from. We actually talked about the folate trap. So all you need to highlight there actually is that 5-methyl tetrahydrofolate there, because you have to move the mouse. And the point I was making there is, if you don't have all the molecules in place, the methionine, the B12 in place in the next cycle, things can get trapped. In other words, they will not move smoothly. So you can have a SNP in that MS gene, methionine synthase gene. You can have SNPs in genes that are important to absorbing and transporting B12. So there's a TCN gene, and one that's very, very important is the FUT. So I call it the FUT2 gene, FUT2 gene. That gene is really instrumental to basically absorbing a distribution of B12 in the body. So you can imagine if you have a SNP there, and I see it all the time, almost always, I see methylation issues with that gene, and also dysbiosis, because it's a gene with a number of different impacts in the body. And one of the things that is, when it's homozygous or it's inefficient, it can deeply impact the bacterial makeup of the gut, which is odd, isn't it? Particularly lactobacillus. So we never, so if I see an aberration in B12, I'm immediately going to start thinking, what's going on in the gut? And that's the power of genomic information. It tells you where to look first. So you're not like, okay, so what's going on with you? Let's just give you some more B12. No, wait a minute. I need to go look at the gut first, because chances are something's off.
Right.
And then I correct that, then I correct this. So that's the folate trap with a little bit more info. Okay, so let's go on to the next slide here. I don't think I have too many more slides. So,
nope.
Oh my gosh, what am I showing? Uh-oh, retro. This is where it starts to get cool. So what I want you to circle is the folate cycle, right? Just highlight the folate cycle. And right next to that, you can see where B12 intersects, right? So we understand that B12 is really important to the cycle. So the cycle right on the left-hand side, which kind of show, is that methylation cycle. What is one of the major products of methylation cycle? Those methyl groups. Circle SAMe there. So SAMe produces methyl groups. Now you're going to take your mouse and go all the way over to that BH4 cycle. And the methyl groups are instrumental to this cycle, which I'll show you in a couple of different ways. So you see MTHFR that I pointed out. MTHFR recycles that BH4 molecule at the top. If you follow the arrows down, you have to do it without me seeing it. Can you see that those orange arrows are basically indicating that BH4 molecule is instrumental to these particular reactions? So it's instrumental to the production of 5-HTP from tryptophan. What's the end product of that, of tryptophan metabolism? Serotonin and melatonin. And see, between serotonin and melatonin, you've got SAMe. What does SAMe mean? What is it giving there? It's giving methyl groups.
Methyl, right.
Right? So can you see the methylation cycle? This is a perfect example of how aberrations in the methylation cycle can impact hormone production. Pretty cool there.
Yeah.
If you also look, follow another arrow down, BH4, the orange arrow to the left, the one furthest to the left.
The L-DOPA.
BH4 is important to driving the production of L-DOPA, precursor to dopamine. So MTHFR, when it gets associated with neurotransmitters, it plays on a couple of ways. First of all, driving the biopterin cycle, and secondly, it gives us those really important methyl groups. So if you look even further down on your diagram where it says norepinephrine at the bottom, circle that.
Yeah.
What you see between norepinephrine and epinephrine? SAMe, right?
Right.
Okay, so circle SAMe, and now everyone should be able to say, okay, SAMe, that stands for acetylmethionine, methionine. Basically, those are those CH4, that's the methyl group. So in other words, to break down norepinephrine into epinephrine, we need methyl groups.
Right.
Which come from the methylation cycle. And the MTHFR plays a role, but as you know, it's only a part of a role. Is it important? Yeah, but there's so much more to consider nutritionally than just MTHFR. That's what I really wanted to show.
Love it.
Okay, on to the next slide then. So I really want to show one more thing. So this is the essential role of COMT, and you're like, okay, what is that? CO-methyl transferase. So COMT is a very, very important gene. It produces CO-methyl transferase. It's an enzyme. What does this enzyme do? Two major things, and I'll navigate this slide in a moment. First of all, it helps to break down estrogen. It helps to detoxify estrogen. Elevated estrogen levels are rampant, right, for many, many, many different reasons. COMT is one of many enzymes that are important in basically breaking down estrogen. It needs methyl groups to be in its active state. So you have to have the methylation cycle working, which is informed by the folate cycle, to create those methyl groups so that COMT can do its work in estrogen detoxification, but also look under here, find dopamine on the chart here.
Mm-hmm.
Go all the way down, it goes to that intermediary DOPAC.
Right.
What does it need to convert DOPAC into homovanillic acid?
COMT.
Bingo. What does COMT need to do its job?
Methyl.
Bingo. You got it? So can you see, and you can see norepinephrine too. So if you look at these neurotransmitters, what they are, so norepinephrine and epinephrine on the right are a kind of fight or flight hormones, right? What happens if we're not able to break them down? What do we hear? People get moody, anxious, jumpy, whatever. They need COMT to break them down. COMT requires methyl groups to do its job.
Got it.
So that's the connection between the methylation cycle. It's one of the connections. I made it very simple. Between the methylation cycle, folate metabolism, and what we hear about mental health. I don't know if anyone can see me. I'm putting mental health in parentheses.
Yeah.
Connection. I don't know if you can see me, but that is the connection, or one of the connections. So we need the base nutrients, right? We need to make our neurotransmitters. We need tyrosine, we need tryptophan, we need our amino acids, but we also need to break them down. And the methylation cycle is really, really important in breaking down these neurotransmitters. Sometimes the COMT, so it's encoded by a gene, right? Sometimes that gene can be sluggish. So we say it can be very slow. And sometimes it can be very fast, regardless of methylation, right? It just is. And so that defines sometimes whether somebody's like super jumpy and anxious or totally like mellow,
Yeah. I think I find it fascinating because I think we spend so much time, especially in the plant versus animal discussion that you were having earlier, but so much time just even focusing on essential amino acids and how we have to get them in, but also not reminding ourselves again, or like the general audience, not maybe getting the reminder clearly that while those are essential amino acids, absolutely, body can't make them, we have to take them in. If we're not getting in these other nutrients properly, the metabolism of those essential amino acids, so it's not just about bringing essential amino acids into the body, they still have obviously healthy digestion to be, is required for utilization, but what you're talking about here is that optimization in accordance with, how you're, like knowing your SNPs and knowing how that part's going to work. And I think that's just so super powerful in the overarching, as you say, like with everything that goes on the plate. You know what I mean? So I think it's really, really important. Yeah.
It is. And, the question I ask myself, so if I'm working with a doctor or I'm working with other clinicians, my colleagues like you, the question we have to ask ourselves is, what are we asking human biochemistry to do?
Right.
And so if somebody said, okay, they're folate deficient or they have a deficiency in something, what we have to do is think, why are they deficient? What is not working? Which requires us to understand biochemistry to know where to look to provide the right fix. And of course, you want to work with food as a fix because it's a natural matrix and information for the human body. As you can see, genes can create some complications where we may need to supplement.
Right.
But we need to be very conservative with supplementation. Know it's a minimal amount of time. Some people may need a lifetime, depending on what's going on. But most of what we need to do can come through food, providing we know what we're doing clinically. So all this to say, just to come from to kind of finish up on the methylation here, is this, I think, is a really interesting, it's called the Rosenberg dopamine bell curve. And all you want to take away from this is what happens when we don't properly metabolize dopamine. So if we don't produce enough neurotransmitters on the left, so the catecholamines are epinephrine, norepinephrine, dopamine. So low levels, you can get food cravings, addiction. That's where substance abuse comes from. Impulsivity. You know, people who are super driven by doing dangerous things, they're doing it. And I just heard Ben Lynch talk with Mike Mutzel about this the other day. A lot of times they're doing it, they're seeking the thrill because they're after the feel good because they have lower levels of dopamine. Right. So impulsivity, you know, we see that with children, like they're all over the place. They can't sit still. They're fidgeting biochemically because they're itching for dopamine. That's one of the thoughts. Whereas the high catecholamines, where it's like they've got an excess. So this may be a case when that COMT enzyme is slow, and we need to find a way to support it, like to methylate it, to get it to do its job, to provide the right substrates so we can get rid of the... Can you imagine if you couldn't get rid of that horrible feeling when you've been frightened or jumpy, right? So you could see where we would get aggression or violence or anxiety or panic.
Right.
When we see that symptomatically, we're going to look at neurotransmitters and say, what nutritionally do we need to look at? What can we support to bring this person down? Because it's not their head, it's their biochemistry.
I think that's also a challenge, too. You can see why clinically medications that also interfere with folic acid, with folate metabolism in the body are so dangerous because they're often used for exactly this purpose, right? So we provide somebody an anti-anxiety or a medication to bring them down, but it's also further interfering. So it's almost taking a deficiency and it's exacerbating that deficiency. So there's a suppression that's going on, but a lack of nurturing. Yeah, that's fascinating.
Yeah, and so that's why it's important to connect everything through these cycles so we can look back and say, well, this cycle feeds this and feeds this. And maybe they do need a bit of more tryptophan or tyrosine, right? It could well be that they don't eat chicken or something.
Yeah. So, this is where I'm going to get on my high horse also. It could also be that your patient is swapping out collagen for protein sources and saying I'm taking in collagen as a protein, and that can be a factor in reducing overall tryptophan. And so that's like it's a super important one to look at. Yeah.
Yeah, no, that's actually a very good point. So it's interesting to look at some case studies and that. So hopefully what I've done here is connect the dots. So I think there's a couple more slides here, which I may just have added in.
Why methylation matters. We've got that slide. But you actually went through this. You talked about, and I'm going to remind people because I'm conscious of time, and also we don't want to overwhelm, but we do have, everybody's going to get this slide. They're going to get this whole thing. So I'm going to hang out on the slides so that they can see them, even if you don't have a comment. Yeah. I think the next two slides are nice to know. So if you kind of zoom to the last slide, I think it says take homes. Yeah. I think, we pushed enough. Important. So there we go. Yes. The last slide. So what are the take homes then? MTHFR is one of many genes that can influence human biochemistry and health. I think if you've learned nothing today, you're going to see that. A combination of MTHFR variants may reduce the efficiency of the enzyme it produces, but may not. Just because you're homozygous, it's not a diagnosis. For clinically, or if you remember the problem, you say, okay, I've got this.
these are the questions I need to look at. Or this is what I need to understand clinically. Some alarm bells should go up. Or no, some not alarm bells, some question marks.
Mm-hmm.
Inefficiencies in MTHFR can be corrected. MTHFR does not work on its own, right? And MTHFR has to be evaluated within the context of many genes and nutrients that are involved in the cycles it informs.
Oh, so brilliant. So brilliant. I'm so happy because when you hear it this way, it's a story. It is super overwhelming. And I just want to invite, we've got, people who have been patiently paying attention and following at every word. So thank you. If you have any questions, this is a great time to drop them in the question box. And I'm going to actually move us over from the slide so that I can be back with Amanda. So let me come over.
Yes, you can see me. Some of you, probably those you listening, you've heard me talk so many times. So
we can see that part, and then we'll drop out of here and we'll pop back up. There we are. Hi, everyone. Oh my gosh, so much brilliance, but also I think if we're unpacking it, you had given these great take homes, and again, everybody's going to get that piece. When somebody is deciding to work with a practitioner, or if you are said practitioner, how do you start? Is it, hey, somebody comes in and the first thing I'm going to do is do this assessment so that I can see, or are you are you doing an overall intake and then then that makes you decide? Yeah.
Oh, that's a question for me, right?
Yes. Yep.
How do I work? It's a very interesting question. Because I work with DNA as my starting point now. You could probably now see why. Because genomic information, and that's my specialty, okay? So not everyone's there yet. But I do that for a couple of reasons. First of all, because it tells you exactly where to look. You've probably seen that today, right? That genomic information, once you know how to interpret it, you create storyboards, and maybe I can come back and show that sometime, Ashley, for our clinicians. What does this look like? Because it's very revelatory. And it tells you exactly what to look at. So you have to understand it. So I will organize the information into tendency to have problems with detoxification, biotransformation, methylation, inflammation, cognitive neurotransmitters. And I was telling, I work with a company in Australia, and I was telling this the other day. I said, when I do that, I actually start with no intake form, which is the reverse of how we've been taught, right? Because what I want to do, and this is how I work, what I want to do is to have no interference at all and have any
no judgment
to anything. I want to read straight DNA, organize it, tell the story of that, and then I bring the patient, because they know I'm going to do this, or the client. And then I said, I've got a bunch of questions for you. When I start asking questions, that's my intake form. But because of the nature of what I'm doing, I also tag team with other clinicians who may or may not be listening right now. Once I've done my DNA and my impression, or I'm working with doctors, this is my impression, these are my recommendations, that's actually when we bring in the intake form. So I use the intake form after I've done my impressions and recommendations to validate what I'm seeing. So the intake form is not what it's not going to predispose me to anything. I actually use it after the fact to validate or to add additional weight to where I might go first with that patient. So if I have somebody who does have anxiety or mood issues or whatever, I'm not like already thinking, okay, well, this is what I'm going to find. I'm going to look in the genes and say, this is what I think is going on. We're now going to do very specific laboratory testing to see if that's right, so I know how to correct it.
Wow, it's like it is totally the reverse, right? And I think what's so great about that part is we have such a tendency, and we, like those people, like, yeah, totally. But we have such a tendency to know information, right? Like, so I know what's right, or I know what's worked, or I've seen this play out. If somebody's got, IBS with constipation, they were on antibiotics, they've got acne, and they were taking birth control. I'm like, okay, I got this story. But to your point, you're not like, it would still be a very difficult stretch for me to not use our digestive evaluation first and just to DNA. Like, I'd be like, oh, I just still need to know about, but I think what you're saying in that space is without any biases at all, let me understand get under the hood and understand what I got to work with. So
I want to look with a blank sheet of paper because we've not been taught to that, and it takes a lot of, I don't say it takes a lot of guts. You have to have confidence in yourself to say, I'm going to go this way first, and then invite you to the conversation, provided the client knows that.
Yeah.
You know, that's how we're doing this.
So the way that you get those guts, my friends, and thank you, because there's no chance that Amanda and I were going to get this done in under an hour. And I had to hold myself back. I had 37 other questions I wanted to ask, which I'll do in our next session. But the way you can get a lot of those answers is she has these amazing courses. And of course, to grab her book, which is coming out September 10th, head to the Genomic Kitchen. And if you liked what you learned here and you want to learn more, give us a thumbs up. You know, if you're on YouTube, you're here on, those things matter. They help us know what content we should be looking at. But most importantly, not about us, is if you have anyone that you think would benefit from knowing this information, we want to be sharing this with other clinicians. We want to be sharing this with anyone who's really in the process of assessment. We talked a lot about mental health here today. There's a lot of, great practitioners who would benefit from being able to have this collaboration with you, the dietitian or the nutritionist, et cetera, in that space. So let's all do better together. And Amanda, always, thank you so much. I just thank you for your curiosity and for leading this. And I think the one of the key things that I'm also learning so much about, especially from you, is this is a space where our global practitioners are doing so much work, and in many instances, again, also not just ahead of the game, but also not encumbered by certain biases. And so one of the things that I look forward to is, you do it on a monthly basis, but I get your update, your email, and it tells me like what to read and what to watch.
Yeah. Kitchen science.
Yeah. You guys sign up for that. There's no way. I mean, the only way that, I look for people who are great vetting systems of other information that's out there so that it's just a super efficiency thing for me. Amanda is one of those. So sign up for her newsletter. Make sure you're getting that so that you get, you know what not to miss, especially. So those are all my recommendations. Thank you to everyone, and we will be getting this out to you in a couple of days via YouTube. I hope you have a great day.
Thanks. Bye, everyone. Thanks for listening.
Yeah.